In our second day of exploration, we learn how our genotype, or our genes, are turned into our phenotype, or what we look like. Now let's talk about the DNA as if it were a recipe book. So inside of each of our cells, we have the full recipe book in the nucleus. Then within that recipe book, we have chapters. So if we zoom in, we can see there's a chapter on appetizers, there's a chapter on breads, there's a chapter on cookies, there's a chapter on candy. These would be similar to a chromosome. So each chapter is a chromosome or a grouping of recipes. And because we are diploid, we actually have 2 copies of every chapter, one from our mother and one from our father. So then if we look at an individual recipe, so let's say we're looking at the mincemeat drop cookies, we would find that we have a full chapter of cookies with a recipe for mincemeat drop cookies from our mother and the same chapter of cookies with a mincemeat drop cookies recipe from our father. And these would represent the different alleles that we have. And so what we want to know is how do we go from the recipe to the finished product or the cookie that we're going to make from the recipe? Well, that process is called the central dogma. In summary, we start with our DNA, which is the original recipe in the nucleus, and we make a photocopy into RNA, which is a close cousin of DNA that we'll talk about in a moment. And then that photocopy is what goes out into the cytoplasm, where it is read by ribosomes and turned into a protein. So we'll talk about each step of this process one at a time. First, let's look at the difference between DNA and RNA. So the DNA, the master recipe in the nucleus, is double-stranded, whereas RNA, the photocopy, is a single-stranded copy of just the coding strand, or just the recipe itself. That's one difference. The second difference is you'll notice the name of the sugar, ribonucleic acid, versus deoxyribonucleic acid, so there's a slight difference in the sugar. The last thing is that in RNA we use a uracil in replace of a thymine. Otherwise they're about identical. Let's start with the process of transcription. So you can see here I've got a DNA molecule. The top strand is running 5' to 3', and the bottom strand is running 3' to 5'. So, given that that's the case, we always read the coding strand from the 5' end. So this would be our coding strand where the gene is, and this bottom strand would be our template strand because it's running in the opposite direction if we are reading this way, which we're going to do. So if we want a copy of the top strand, we're simply going to use the template or the bottom strand to base pair to. So if I see an A, I would write a T, but in this case I'm using RNA, so I'm going to write a U. If I see a T, I write an A, U, A, G, C, U, G, U, A, etc. And you'll notice that if you look at the sequence of this strand that we've just built, it looks exactly like the top strand, except U's instead of T's. And this particular molecule we've just built is called a messenger RNA, and it's built by the enzyme RNA polymerase because it's building an RNA polymer. The next step is RNA processing. There are 3 things we have to do. The first is to remove any of the junk that we call introns and keep the exons. So basically, we're going to splice our piece of mRNA apart, and we're going to only keep the parts that were introns. We'll keep that part, and we'll stick them together. The second step is to add a 5' cap. So we'll just call that this little guy right here. Here's our 5' cap. And the last step is to add a poly-A tail. So a whole bunch of A's to the end. This is now a processed mRNA. The last step is translation. At this point, our processed mRNA that we have here gets attached to the ribosome out in the cytoplasm, and it gets read. So we're going to have transfer RNAs bring in amino acids depending on what their anticodon is. So if this transfer RNA is going to attach to AUG, which is the codon, it would read UAC. And if we look at the code, we have to find AUG. So first letter A, second letter U, third letter G. Gives us right here. So that means that methionine is going to be that first amino acid. Then we'll get another transfer RNA coming along, and it's going to match the codon CAC. Let's label that. This is our codon. And up here on the tRNA is the anticodon. And in this case, it's going to be GUG. And CAC, if we look at our code, CAC is a histidine. So a histidine will be the next amino acid in the line, and so on and so forth to the end. These connect together to make a polypeptide, or a chain of amino acids. And once that's done, we have finished the process of reading our DNA and making it into a protein.